These are the lecture notes for Chapter 5, which is on the macromolecules or the large biological molecules. And there are 4 groups of those. The 3 of them are like food groups. So there's carbohydrates, proteins, lipids, and nucleic acids. When we talk about the 4 groups of biological molecules or macromolecules just means big molecules. So every one of them has substances in that group that are small, and they're considered building blocks or monomers. And then substances that are very, very large, long chains of monomers, and we call those polymers. So, for example, proteins, you may even know this, that proteins are composed of building blocks called amino acids. So you would say the protein is the polymer and the amino acid is the monomer. And we'll talk about more. But anyway, in your notes, make sure you know that macromolecules just means big molecules. There are 4 groups of macromolecules. And for each group, you have polymers, which are large, long chains of smaller building blocks called monomers. And the monomers are the building blocks. And the macromolecules that we consider to be polymers or long chains are the carbohydrates, proteins, and nucleic acids, because the lipids, even though they are large, they're not really technically polymers. Okay, so specialized proteins or macromolecules that speed up chemical reactions are enzymes. So they are protein catalyst, basically, protein catalyst. Enzymes speed up chemical reactions. And we have 2 basic types of chemical reactions. Make sure you know this because I know for sure this is one of your questions on your next exam. A dehydration reaction is a synthesis reaction. That is when you build a larger molecule from 2 small molecules. For example, when 2 monomers or building blocks chemically bond together to build a larger molecule, you call that a dehydration synthesis reaction. But then we have breakdown reactions called hydrolysis, which are the reverse of dehydration. Okay, so here we're looking at the dehydration and hydrolysis reactions of proteins. So when we are synthesizing a protein, a dehydration reaction would look like this. You would have a short polymer already. So these molecules 1, 2, and 3 are amino acids. They're going to be joined with another monomer or amino acid. And when they're joined together, a water molecule will be removed, and that's why the term dehydration is used. So it's synthesis because you're building a longer polymer, and it's dehydration because a water molecule is removed in the process. Notice that water is made of 2 hydrogen atoms and 1 oxygen atom. So one of the hydrogens is removed from the short polymer, and then the OH is removed from the monomer. And when they are removed, that kind of leaves room for a chemical bond to form between the 2. So then you have a longer polymer that has 4 amino acids in it rather than 3. And that is how polymers are built, by dehydration synthesis reactions. And then the breaking down, instead of removing a water molecule is added. So hydro is water, lysis is breakdown. It's the breakdown of a larger molecule through the removal of water. And so dehydration is synthesis, hydrolysis is breakdown. The video that you're looking at is on slide 7 if you want to watch a video of how polymers and monomers are produced and broken down. And we're going to start with the first group, which is the carbohydrates. And you probably know from nutrition what carbohydrates are. They're going to be sugars and starches, or what we call simple carbs and complex carbs. So the simplest carbohydrates are monosaccharides, or simple sugars. And probably the most common sugar is actually a disaccharide called sucrose, or table sugar, which is not a simple sugar, but it's still a sugar. And it's still a small carbohydrate. But the monosaccharides, that literally means, mono means one and saccharide means sugar. So the monosaccharides are the simple sugars. For example, glucose, which is our blood sugar. And then the macromolecules or polymers of carbohydrates are called polysaccharides. And the one that we're probably most familiar with is starch, like you find in bread and potatoes and pasta. So a polysaccharide is many chains of simple sugars or glucose. All right, so our monosaccharide, the most well-known one is glucose, and here's the chemical formula for glucose. You do need to know this, not just for this chapter, but also for cellular respiration and photosynthesis, which are chapters that are coming up. So glucose is also known as blood sugar, but it's the most common monosaccharide, and the chemical formula is C6H12O6, and that means 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. And I will tell you, just so you'll understand the table here, some of these sugars are called aldoses and some are called ketoses, and that has to do with, there's this part of the molecule that is a carbon double bonded to an oxygen. And so when that carbon double bonded to oxygen is on the end of the molecule, we call it an aldehyde. When it's in the middle of the molecule, we call it a ketose. Okay, so we have ribose, which is the sugar found in RNA. Deoxyribose is the sugar found in DNA. And those are aldose sugars. They have 5 carbons, so we can also refer to them as pentose. Pentose for 5. And then we have the 6 carbon sugars like glucose. Glucose and galactose, which is a monosaccharide found in milk. Those are aldose sugars, and fructose is a monosaccharide found in fruit that is a ketose sugar. Okay, here is a video, and this is from slide number 12 if you want to watch the video on the synthesis of sucrose. Sucrose is a sugar, a disaccharide that is composed of glucose and fructose. And I'm going to ask you a question real quick just to see if you understood about the dehydration and hydrolysis reactions. So what would the reaction be called that joins glucose and fructose to make sucrose? And since it is a synthesis reaction, that would be a dehydration reaction. This is just the structural formulas for glucose. And then again, disaccharides are 2 monosaccharides joined together. Sucrose is the most common one of those. So sucrose is composed of the monosaccharides, glucose and fructose. And maltose, that is the sugar that's found in, like you may have had, it's a candy that you can get at the movie theater. I guess you guys go to movie theaters still. Malt sugar is found in like the candies called Whoppers. They have that kind of, it's malted milk hardened in the center that's surrounded by chocolate. But another place is in milkshakes that have malt powder added to them. So you would call it, like if you get a chocolate milkshake and it has malt powder in it, it's called a chocolate malt. So that is an example of where we find maltose sugar, and also in malt beverages like malt liquor. There's a video on monosaccharides. This is slide number 17 if you'd like to watch that. And then we have polysaccharides, and we have a lot more examples of polysaccharides in nature. These are all going to be chains of glucose, the monosaccharide glucose. So the polysaccharides are the polymers of carbohydrates. Plants store glucose as starch, so it's a storage polysaccharide in plants. And then animal cells store glucose as glycogen, and we typically do that in our liver cells, and our muscle cells. So glycogen is kind of like starch, but it's not found in plant cells, it's found in animal cells, and it's also found in fungi. Believe it or not, fungi like mushrooms are have a lot more in common with the animal kingdom than with the plant kingdom. You may not see that, but when you learn more about them, you will. So fungi also store glucose as glycogen rather than starch, similar to animal cells. So glycogen is stored mainly in animals, in the liver and in muscle cells. Okay, so glycogen is the storage form of glucose in animal cells, and starch is the storage form in plant cells. Plant cell walls are made of a polysaccharide called cellulose. So the cell wall is outside of the plasma membrane. You may remember that from lab. The cell wall is outside the plasma membrane. We labeled that in the onion cell. So that cell wall in plants is composed of cellulose, which is also a polymer of glucose. And what else have we got here? There's a video on polysaccharides, slide number 25. And I'm going to skip some of this because it goes into a little bit more chemistry than I am requiring you to know. Something about cellulose is what in nutrition is what we call insoluble fiber. So we get it from eating salads, eating plant foods, particularly that hasn't been cooked, raw plant material. A lot of animals that are called herbivores, like cows and horses, and even termites, which are insects, of course, have microbes that live inside of their gut, inside of their intestines, that can break down the cellulose in grass or wood, and that's how they're able to digest it. But humans cannot digest cellulose. We do need it, though, to help us with digestion. It helps our food to digest, to move through the intestinal digestive tract easier. Now, another polysaccharide is called chitin. Chitin is the material found in the cell walls of fungi. And then we also have cell walls in bacteria. And that one's not on here, but that chemical is called peptidoglycan. Peptidoglycan. That's a polysaccharide found in the cell walls of bacteria. And then as you see here, chitin is found in not just the cell walls of fungi, but also the exoskeletons of arthropods. And here you have what looks like a locust, emerging and leaving behind its exoskeleton. So we call that molting. All right, our next class, I'll try to get this finished before I have to stop the recording. But our next class of macromolecules are called lipids, and they don't form true polymers. They form large molecules for sure, but they consist of hydrocarbon chains. That's literally what it sounds like, hydrogen and carbon. Hydrocarbon chains, and then a chemical called glycerol. The most important lipids are fats and oils, and then the phospholipids that make up our plasma membrane, and then steroids, which form hormones like testosterone. So fats are constructed from, and fats can be liquid or solid, but they're constructed from a molecule called glycerol, and then those hydrocarbon chains form fatty acids. A fatty acid is a hydrocarbon chain that has a carboxyl group attached to it, and I'll show you what I mean. It's a carboxyl group attached to one of those hydrocarbon chains, which forms a long carbon skeleton or chain. So let's look and see what that how that looks. So down here at the bottom, this is our triglyceride, which is human fat is almost always triglyceride. So this molecule in gray is a glycerol molecule, and then attached are 3 fatty acid tails. And you can notice for every fatty acid tail, it's a hydrocarbon chain attached to CO, and it would be an OH, except the H is removed when it binds with the glycerol. So look up here and you can see it. The COOH is the carboxyl group. COOH, sorry, I don't mean to highlight over here. I just mean the COOH is our carboxyl group, and that's what makes the fatty acid an acid. So fatty acids have a long hydrocarbon chain attached to a carboxyl group, and then you have glycerol. And those that glycerol is joined to 3 fatty acid tails to form a triglyceride, which can either be liquid or solid, so it's a fat or an oil. And this is slide number 34, and this is where we'll start the next recording, is we'll talk about the differences between saturated and unsaturated fatty acids.